Thanks. This is great. It's the last year of the cluster, and it's exciting to be able to present all the results that we found during this project. 5 years is a long time for a project, and we've been able to really dig deep into this issue of how we can change certain crop production practices to help get better pest control in greenhouse ornamentals. Before going into the details, I'd like to present the team. The research team, of course, Sarah and Siobhan are very important to this project with their connections to the real world. Whatever we do in our research greenhouses, they can translate that to what's actually happening and important in commercial production. Also, our technicians on this project, Ashley, Jesse, Lakshmi, and then for the last 2 years, we've teamed up with the biochemistry team here at Vineland, Dr. David Liscombe and Dr. Christine Kempthorne. So very exciting collaborations here. So I'll first give a little bit of background for those of you who are not familiar with the project, why we are looking at these things. The whole strategy really was to help growers who are already doing a lot of biocontrol, make their biocontrol even more successful. Basically, 94% of floriculture growers in Ontario are using biological control, and it's their primary IPM strategy. So this infographic is a summary of all the strategies that we have done research on over the past 10, 15 years. This project really looked into the use of cutting dips at the beginning. You see the little leaves submerged there, and also the effect of plant resilience. How can we make the plants more resilient? And the theory behind this is this. This graph is a theoretical graph. What happens to the number of thrips on the y-axis over time if you do certain things. Now, if you do nothing, the red line, basically thrips will increase and get out of hand. These cutting dips will knock down pest populations and actually win time for the bios to start working. And then you get the green line. But because it happens at the beginning, if you don't do anything else, eventually thrips will increase again. Which is why we were looking at plant resilience. And it means finding a way to make the plants less susceptible or more resistant to pests. Because when plants are less susceptible, you slow down their population growth. And this is the blue arrow with the blue line. And this makes it easier for biocontrol agents to manage pests. And one of the ways this could be done is by optimizing plant nutrition to benefit the plant, as Barry has shown, but not the pests. And there was some previous research already done when we started this project that showed that reduced fertilizer likely lowers the plant's nutritional value to pests or could increase plant defenses. And in these graphs on the y-axis, it's always the number of thrips. And then you can see that at different fertilizer levels, which are the different treatments shown here, there's less pests, less thrips at lower fertilizer levels. And in the most right-hand graph, you can even see that there's better biocontrol performance. So there was some precedent before we started this project. However, we started thinking as well, if you lower fertilizer even more or too much, of course, it will negatively impact plant quality. I'm very happy that Barry was able to show that nutrients can be reduced a lot in floriculture without any negative effects. But if there would be negative effects, we thought biostimulants could be of help. Biostimulants are beneficial bacteria or fungi or extracts that can mitigate these negative effects by helping the plants deal with nutrients. At the same time, we know that several biostimulants can actually induce the plants to produce defense compounds or release volatiles that attract natural enemies, as you can see in this picture, which can also reduce or slow down pest populations. So that's why we thought to combine biostimulants with the reduced fertilizer. So overall, the objectives of this 5-year project were first to eliminate thrips on imported cuttings by cutting dips, then optimize nutrient inputs and biostimulants to reduce the risk of pest outbreaks, with the goal of still maintaining plant health and quality for gerberas and chrysanthemums, and also look at the effects of reduced fertilizers or biostimulants on biocontrol efficiency. Then the plan was to validate these effects that we hopefully would find in 2 commercial crops from start to finish, incorporating kind of our learnings from the previous years and compare it to conventional growing practices. And then, of course, do a lot of knowledge transfer and outreach activities to actually make the users, the growers, aware of our results. So to recap really quickly the results of year 1 to 4, I've presented this before, so we'll go through this really quickly. We found, of course, that it was clear that dipping unrooted cuttings reduced thrips populations and increased the success of an IPM program. Now, with regards to optimizing plant nutrition, to reduce the risk of pest outbreaks, we tried it first on potted gerbera with whiteflies. However, we did not see any effects and any meaningful effects on the whitefly populations within the time frame of a short potted gerbera crop cycle. And this is probably because whitefly population development is pretty slow, actually. Their life cycle is really long. We also did not find any effect of reduced nutrients and host plant quality on the susceptibility of whiteflies to a fungal biopesticide like Botanicard. So that's in gerbera and whiteflies, we did not find any reasonable effects. However, in chrysanthemums with thrips, we did find that when we did these trials, a lot of trials in summer and winter over the course of the years, when we took all these greenhouse trials together, we did see a possible effect of plant nutrition on thrips populations. And we saw that there could be a possible reduction between 30 and 50% of a whole fertilizer recipe. However, as you can see in this picture of all these chrysanthemum plants at different rates, when you lower fertilizer rate, in our case, we often saw that the lower fertilizer plants, and here I refer to like the 50 ppm or the 100 ppm, there were some negative effects on plant quality. So that's where we thought, let's get in our biostimulants. So the first batch of biostimulants that we tried did not mitigate the effects of low fertilizer, and it did not seem to induce any plant defenses against thrips. Also, when we combined the lower fertilizer rates with thrips biocontrol, we actually saw no differences in thrips populations, probably because our biocontrol program was actually really strong. So it worked no matter what our plant susceptibility was. So at the beginning of last year, so year 4, with these results, we were not yet ready to really go into commercial greenhouses and test our results. So we decided to rethink the project and actually look more into what was happening in the plants, because we saw a lot of variability in our results, and we thought that this variability may be due to plant species or variety, the environment, temperature, summer, winter, substrate and fertilizer type. We also didn't know really what was happening in the plant. We just put fertilizer and then we looked at thrips. So anything that happened in between, we had no idea. And of course, maybe different biostimulants products could work in a different way because there's so many different products out there that you can try. So we decided to focus on thrips oviposition in the lab. And we grew the plants either in a growth chamber or in the greenhouse. And then we took leaves back to the lab and put thrips on them and then counted how many eggs they laid. And we looked at thrips damage, their feeding, combined with plant metabolite analysis. So this is where we teamed up with the biochemistry team and took plant samples and really analyzed all the secondary metabolites in the plant to see what was actually happening in the plants. And I will present several of our experiments in the next few slides. So when we looked at chrysanthemum, we varied fertilizer rate, of course, high and low. The high rate was sufficient. The low rate was like that borderline kind of rate, 75 ppm of nitrogen, where we saw some effect on plant quality. And then we looked at 4 different varieties because in previous trials with other plant species, we saw that variety actually had a big effect on how plants actually reacted to pests. So we repeated this trial 11 times. Thank you, Jessie. And the results, and we did this so many times because the results were not consistent among trials. The graph with the green bars, it's the number of thrips oviposition in the different cultivars. And this is all taken together. But when you look at the individual trials, there sometimes was an effect of low fertilizer plants, that they have less thrips oviposition, but other cases we did not see this effect. So this graph mashes everything together and kind of models the picture a little bit. We also saw that several plant metabolites, for example, related to plant defenses like chlorogenic acid, and that's the graph on the right, did vary according to fertilizer rate. So actually the low fertilizer plants had more plant defenses than the high fertilizer rate. So what we actually need to do now is take all these results and do a meta-analysis to correlate thrips oviposition with the metabolites, with the SAP analysis we did, time of year, environmental conditions, etc. So a whole bunch of statistics need to happen to actually tease out when low fertilizer actually leads to less thrips oviposition and when it doesn't. However, this is more biochemistry analysis. We did see that actually there were big difference among the 4 varieties that we tested. And how to read these graphs is basically we grouped the different samples together. So when dots are closer, that means they're more similar. And imagine a whole cloud in a room where these samples are grouped, and then we just smash it into 2 dimensions, into a flat plane. And this is what you get. So the different colors, CH, GP, VY, and SP, those are the 4 different plant varieties. And you can see that they are very different from each other. The different colors have light and dark dots as well. And you can see those are really close together, and that's the high and low fertilizer. So there's way less differences due to fertilizer than due to variety. I hope I explained this well. So in the next experiment, we try to take this a little bit further. We varied fertilizer, high and low. We had 2 plant varieties, and we added biostimulants, a different batch of products than we tried before. To summarize this huge experiment, we did see differences in fertilizer. due to the effective fertilizer rate. So in this graph, you can see that in Grandview and environment, we see that the low fertilizer plants had less thrips than the high fertilizer plants, which is according to our expectations. But we did not see any differences due to biostimulants in thrips numbers. Also, we saw, again, these differences between varieties. Apparently, Viren is more, it's a better host plant for thrips than Grandview. And the lower graph, it's thrips damage, which was actually the opposite. Although Viren is very suitable plant for thrips, we saw less damage on them. But again, the low fertilizer plants in Grandview had less damage than the high fertilizer plants. Also in the plants, the biostimulants did not do much for like shoot growth and root growth. The main differences were really related to fertilizer rate. The last thing we did was in this experiment was to look at what was different in the secondary metabolites. And in that cloud graph, you can see that the low fertilizer and the high fertilizer plants did kind of group separately. So there were differences between the low and high in this variety Viren. And at the bottom, you can see several graphs that are related to the different metabolites that we found. So related to plant defenses, the chlorogenic acid, we see again that the low fertilizer plants have more defenses than the high fertilizer plants. The 4 graphs on the right actually show that there's less amino acids in the low fertilizer plants and the high fertilizer plants. So that means that there's less food for thrips to eat. So these plants were less suitable to thrips. So more defenses and less food in low fertilizer plants, which kind of explains why we saw less thrips in the low fertilizer plants. The results for the other variety, Grandview, are still pending. We're hoping to get those in a few weeks, and hopefully they will confirm these results. But if we look at also what happens if we put thrips on these plants, we saw that, again, there was no induction of plant defenses by the biostimulants, but the low fertilizer plants definitely had a higher defense reaction upon thrips feeding than the high fertilizer plants. So there again, there is a difference in induced defenses against thrips as well. And again, the results for Grandview, we're still waiting for those. So that was chrysanthemum. So apparently biostimulants and chrysanthemum, they go like, we don't care. So we tried it a second time, but this time in Gerbera. We repeated this fertilizer and biostimulants trial in Gerbera, and the results were slightly different. We haven't finished analyzing this as well, but what we can say is, again, we saw less thrips oviposition in the low fertilizer plants compared to the high fertilizer plants. And there was a slight effect, subtle difference due to biostimulants. So maybe something happened there to induce defenses or not. But overall, more eggs were laid on the high fertilizer plants. And we also saw in the bottom graph, slightly more thrips damage on the low fertilizer plants in this case, or no difference, depending on which biostimulant we use. So I don't really have a good explanation yet for this, but I think the differences are actually very subtle. So there may be a difference if you put it in a graph, but if you would look at the whole plant, it may not be very apparent. And again, the metabolite analysis for the Gerbera is pending. So hopefully we will be able to pick apart the different effects based on what's happening in the plant. Instead of a commercial trial testing all these different things, we decided to do a trial just looking at the different effects of variety in a commercial operation. So we went to Sawaya, Mel Sawaya, and he was doing these garden mom variety trials, and he allowed us to look at how many thrips we actually found on different varieties of plants. And we very conveniently called them A, B, C, D, E, F, because it doesn't really matter what their names are. But what matters is that they're all yellow garden mums. They all look very similar. However, in these graphs, you can already see that during the vegetative stage or the budding stage, there are differences between these yellow varieties. Some have more thrips than others. In the greenhouse, we saw mostly Western flower thrips. Outside, onion thrips came into the mix. But overall, variety A, D, and F had very low thrips numbers, which would be very good varieties then to choose if you want to avoid having high thrips populations. Variety B and C had the highest thrips numbers, but on B, over time, when the plants were budding, these numbers were really low. So actually only variety C stood out really high in the budding stage. And then once plants started flowering, of course, thrips are very much attracted to flowers. So that's where we saw most of the thrips. When we took samples for the metabolite analysis, we saw again that the profile of variety C was very different from the other varieties. And again, this was related to amino acids, so thrips food content, and plant defenses, the chlorogenic acid. And many metabolites remain to be annotated. We're not done analyzing all the different metabolites yet. But again, it shows that varieties can differ very much in how they react to thrips. So in conclusion, I think we've seen that lowering fertilizer rate may very well reduce losses due to thrips. It's still a bit of a question, like under which conditions this happens and under which conditions this does not happen. So we still need to further untangle all these different factors affecting plant resilience. It was a bit of a surprise that the tested biostimulants did not compensate for reduced fertilizer or induced defenses in chrysanthemums. But, oh well, maybe there are other products out there that will do this. I don't know. I think the main message really is that the selection of resistant cultivars is very important, because the effect of cultivar was much bigger than the effect of fertilizer rate. And then if you have to grow susceptible plants, you can concentrate your bio program on those more susceptible plants. But overall, I echo Barry's conclusion that reduction in fertilizer applications is possible and will also lead to direct cost savings through reduced inputs. So because we can never say no to a very quick additional trial, here's a little encore for dips for tropical thrips. We had the opportunity to actually work with a new thrip species in Canada, Thrips parvispinus, and which comes in on tropical plant cuttings. And this graph shows you real quick that actually the dips that we tested for Western flower thrips will also work very well on this thrip species. So the dips in botanic art or mineral oil will reduce Thrips parvispinus by 70%. Overall, we talked a lot about our work in at least 5 presentations this year, and we are also going to write at least one scientific paper once we analyze our last results. We still have a little bit of funding from a different funding source, the American Floral Endowment, to continue this work for a few more months. So hopefully we'll be able to wrap a nice bow around this project. And it was a blast. It was really fun. So I want to thank everyone at the Biocontrol Lab, the Violent Greenhouse staff, and also Gaetan Degle for statistical analysis, all the funders. And here's my email address if you want to ask any questions, if you can't ask them right now, because I know I went on a little bit longer. So thank you very much.
Thank you, Rose. And I will open it up for one or two questions. We're a bit behind, but got all day to make it up again. I don't see any yet, but my I was interested in your comment that your thrips biocontrol program was working too well for you in this trial, and you didn't have enough thrips, and it shows me some of the incremental achievements we've made through a lot of this research over the years. When the cluster started, the dipping work was just beginning, and you were helping growers develop thrips biocontrol programs that are very, very effective these days, with many growers having systems that don't rely on chemical pesticides anymore at all for Western flower thrips. So anyway, that's a neat aside I just wanted to highlight. And we've come a long way since those days in terms of the research that you've done.
We never rest,
of
because now we
controls.
have other thrips species that we have to deal with as well, and maybe tweak our biocontrol program a bit. So that's the next
Absolutely.
frontier.
Yeah, it doesn't stop. So anyway, thank you, Rose.